Hello, and welcome to the Wiley Web Seminar, Mass Photometry to Study the Oligomerization of HACE1. My name is Christine Smith, and I'm an editor with Wiley's Corporate Solutions team, and I will be moderating today's event. This web seminar, led by Sonja Lorenz from the Max Planck Institute for Multidisciplinary Sciences and Jan Schäferkort from Refine, highlights the use of mass photometry when investigating HACE1 regulation and its substrate specificity. As an independent group leader at the Max Planck Institute for Multidisciplinary Sciences, Dr. Sonja Lorenz supports researchers in the field of biochemistry and molecular biology. She has been actively researching and teaching about the ubiquitin system, with particular focus on HECT-type ubiquitin ligases for the past 20 years to accelerate the development and optimization of biochemical processes and technologies. She received her PhD in biochemistry from the University of Oxford, completed a postdoctoral fellowship at the University of California, Berkeley, and has been an Emmy Noether group leader at the University of Würzburg. Since 2021, she has been leading her own group at the Max Planck Institute for Multidisciplinary Sciences in Göttingen, Germany. Welcome, Sonja. Dr. Jan Schäferkort obtained his PhD at the University of Dusseldorf in the Department of Physical Biology and started his life science career 22 years ago with commercializing microarray technology and services. With technical expertise in molecular biology, cell biology, automation, and project screening, he has amassed a fantastic overview on molecular and cell analysis and how it has developed in this time. As technical sales manager for Germany at Refine, he develops the new and exciting mass photometry market to help accelerate science by making sample characterization more straightforward. Welcome, Jan. And with that, I now hand it over to Sonja and Jan and look forward to their presentations.
Hello, everyone. I'm very happy and pleased to kick off this session with an introduction into mass photometry. In the next few minutes, I would like to show you the principle, how mass photometry works, and to prepare the stage for Sonja. Molecular mass is a universal readout that informs on sample homogeneity, on structural integrity, and activity of biomolecules. It is important to understand that mass photometry is a single particle method which measures the biomolecules in their native state and without needing any labels. It measures the light scattered by individual particles and uses a signal to count the particle and measures their mass. Mass photometry is versatile because it measures mass, which is a universal property. This versatility is a major strength because mass photometry can provide useful information on many different types of samples, including proteins, nucleic acid, and complexes. It is the perfect method to be used in quality control, for example, before moving to much more time-consuming and more extensive or sample-intensive methods like cryo-EM or biosensoric methods like SPR or others. The slide gives you an overview for the different applications mass photometry is useful for. So the overall topics are sample characterization, which includes the measurement of aggregation, effects, fill ratio if you think on AAV empty full ratio measurements, or just sample quality control if your sample is homogeneous. You can do interaction studies, including binding affinity measurements, assembly mechanisms, or you can get an idea of the complex stoichiometry. But you can also do analysis on the complexes itself, means you can look to the oligomeric state or complex composition. Mass photometry has a wide working range, enabling you to reliably measure molecular mass in the range from 30 kilodalton to 5 megadalton, and is covering nucleic acid analysis, proteins or their complexes, or even small viral particles like the AAVs I mentioned in the slide before. Everything within this mass range can be detected and analyzed with a sensitivity which is lower than around about a nanogram if we are talking about proteins. So how does mass photometry work? What do we need to measure a sample in a 2MP? So we need super clean glass slides and silicone cassettes, which just give you the space to pipette independent samples on one glass slide. So usually for manual measurement, we take cassettes with 6 holes to measure 6 samples on one glass slide. So where do we measure? We measure on the glass liquid interface. So this is a glass slide, this is a droplet with your biomolecules, and we measure in this glass liquid interface. Once a molecule is attaching the surface, this is a moment of measurement where we can detect those point spread function. How does it work? We shine light from the bottom to the top of the glass slide, and most of the light is reflected. But then we also have scattered light, which is scattered when the molecule is attaching the glass surface. We take the constructive interference signal from the scattered light and the reflected light, and this will cause those relative contrasts, which are called point spread function. These point spread function, or relative contrasts, are linear proportional to the mass of the molecule in the range from 30 kilodalton to 5 megadalton. The typical plot you might have faced before when watching a mass photometry result is we have the counts on the y-axis and the relative contrast or the mass on the x-axis. So this is how mass photometry works. Mass photometry is very powerful for the quick analysis of your samples with single molecule resolution. The procedure to prepare your sample is just a dilution to nanomolar concentration. With 10 to 20 microliter of volume, the sample consumption is round about 1,000 times lower and round about 10 times faster compared to size exclusion chromatography. To give you a quick overview about the workflow, I brought a video with me where my colleague is doing the experiment on a 2MP. So here's the optical unit and there's a lid. You have to open the lid, and here's the stage with the glass slide and the silicone cassette on it. He just put the sample on it. Now he takes another pipette to dilute it further. After dilution, he's closing the lid and is starting the video at the instrument. The video is taken and recorded for 1 minute, and here you can nicely see the point spread function in that video occurred by the molecules attaching to the surface. Summarizing the specification of the 2MP, we are working in a mass range between 30 kilodalton and 5 megadalton with a standard concentration of round about 10 nanomolar, whereas 10 to 20 microliter is needed. We have a baseline resolution of 2 independent proteins if their mass differs round about 25 kilodalton for a 66 kilodalton protein or 60 kilodalton for a 660 kilodalton protein. As the mass precision is 2%, we can differentiate also proteins with much lower molecular mass difference. I would like to finish my introduction with an overview about our instrument range or the mass photometry family. So this is a 2MP and the 2MP Auto. So we do not only have the manual system to do the analysis, we can also give you it in combination with a robotic system, which is doing the dilution and the application of the sample to the glass surface. With the automated solution, you can measure up to 24 samples without hands-on time. This is also possible with the SAMUX. The SAMUX is our dedicated system for the AAV analysis. Also, the SAMUX can be integrated into the robotic system. So the systems, the 2MP and the SAMUX, they are placed on the bottom of the automation system. Our latest child in the family is the Caritro. The Caritro is working in the macro mass photometry range, which is a bit different to mass photometry. What do you measure with the Caritro? You're measuring the size, and you can measure particles between 40 nanometer and 150 nanometer, and you measure the contrast, which is a proxy for the mass. So you have two information you receive from the Caritro quite quickly to do sample characterization on bigger viral particles like lentiviral or adenoviral particles. So last but not least, I would like also to refer to our homepage where you can get further information, and our YouTube channel. If you want to have more dedicated information on some topics, it's worth to have a look at the YouTube channel. And at the end, I would like to thank you for your attention, and I move to Sonja. Thank you.
Good afternoon. My name is Sonja Lorenz, and I'm a group leader at the Max Planck Institute for Multidisciplinary Sciences in Göttingen. My lab is interested in the structural mechanisms of post-translational protein modifications, and we're specifically fascinated with the ubiquitin system. What is ubiquitin? Ubiquitin is a small protein modifier that programs the fate of its target proteins in countless ways and countless cellular pathways. Ubiquitin has 76 residues, 8.5 kilodalton, and the fold you can see here, it's called a beta grasp fold that is flanked by a C-terminal flexible extension by which ubiquitin is attached to its target proteins. Ubiquitin has been highly conserved across hundreds of millions of years of eukaryotic evolution, as you can see here from the sequence alignment, just showing how important and how critical the fold and the surfaces of ubiquitin are for cellular signaling. To understand this versatility and specificity of ubiquitin in programming so many different pathways, we need to look at the cascade or the catalytic machinery that drives ubiquitination and the attachment of ubiquitin to target proteins. This cascade involves a regulated sequence of protein-protein interactions. First, ubiquitin is activated by an E1 enzyme. This involves ATP hydrolysis and generates an energy-rich thioester between the C-terminus of ubiquitin and the catalytic cysteine of the E1. This thioester is then converted into another intermediate and another thioester-linked complex, and now with the ubiquitin conjugating enzyme E2. And this complex then interacts with members of the E3 family of ubiquitin ligases in order to transfer ubiquitin over to substrates. This step can follow different mechanisms depending on the class of ligase involved. The majority of ligases are so-called ring-type ligases, which function as catalytic scaffolds that bring a ubiquitin-loaded E2 and a substrate into proximity and catalyze the direct transfer of ubiquitin from the E2 onto the substrate. In contrast, other E3s, such as HECT and RBR type E3s, have a catalytic cysteine themselves, take over ubiquitin from the E2 in the form of the thioester intermediate, and then catalyze the transfer over to the substrate in a second step. So substrates may not only be modified with one ubiquitin molecule, but with polymeric chains that can adopt different, can have different linkage types and adopt different conformations. And these occur through iterations of this catalytic cascade that's drawn here. Proteomic studies showed that there are over 50,000 sites, ubiquitination sites in the human proteome. There are 8 different linkage types that chains can contain. And of course, these chains can also have mixed linkages and therefore adopt different topologies. So there is an enormous diversity of ubiquitin modifications that can be formed on substrates and program distinct cellular outcomes for the modified substrate. The best known outcome of ubiquitination is proteasomal degradation. However, there are many other fates and possibilities, such as changes in localization, changes in structure or conformational dynamics, changes in the composition of complexes, or changes in activity of the modified substrates. Ubiquitination is also a dynamic process due to the action of deubiquitinases, short dubs that can cleave and edit ubiquitin modifications and therefore render the system reversible and even more complex. Now, if we look at the multiplicity of enzymes in this cascade, it's very clear that the most versatile family and therefore also the most specific family are these E3 ligases, of which several hundred proteins exist in the human proteome. And as a lab that's interested in understanding specificity of this system, our main focus is on understanding the actions of these ubiquitin ligases, specifically their interactions with the upstream E2 enzyme, with ubiquitin substrates, with the target proteins, and with regulatory factors that modulate their activities. Specifically, I want to show you today data on a HECT-type ligase. So this is a catalytic cysteine-containing ligase from this family down here. The HECT family has 28 members in the human proteome, which are shown here, and they are all characterized by a C-terminal catalytic domain. While the N-terminal regions are highly diverse, they are responsible for the recruitment of specific substrates and regulators, and they mainly drive the specific functions of these members. Specifically, I will talk about HACE1 today, which is a ligase that is very important in the regulation of membrane dynamics, for example, during the maintenance of the Golgi apparatus or autophagy, as well as cell migration. Loss of HACE1 or loss of function of HACE1 is tightly linked to neurodevelopmental disorders in humans, so it's a very interesting enzyme to study and to dissect the mechanism of. Now, the data I'm going to show you now and that ought to feature the use we make of mass photometry, as well as other techniques, of course, that we're using, are derived from a recently published story that is a much more comprehensive story than what I have time to show now, but I'm going to now extract certain data from this paper to highlight how we combine techniques to study oligomerization of HACE1. So when looking at full-length purified HACE1, this is a 100 kilodalton protein. We saw by size exclusion chromatography that the protein elutes as an oligomer at higher apparent molecular weight than expected. And we could use mass photometry here to pinpoint that this peak corresponds to a dimeric form of the enzyme. This dimerization is independent of the system which we use for the recombinant preparation of the protein. We are comparing here protein from E. coli, from baculovirus insect cell expression, and from mammalian cells. And you can see all these proteins are fully dimeric. We went on to determine a cryo-EM structure of this dimeric state, which revealed an intriguing yin-yang type dimeric arrangement, in which the interface, interestingly, here between the two subunits contains the catalytic heck domain. We also studied the conformation of HACE1 in solution using small angle X-ray scattering, short SAX. And I'm showing you here a superposition of the experimentally determined scattering data in cyan and a simulated curve that was calculated based on this cryo-EM structure. And the very good correspondence of these two curves really shows that the shape and the geometry of the HACE1 particle in solution resembles the cryo-EM structure we had determined. Now, looking closer at the dimerization interface, you can appreciate here this is composed mainly of an interface between the N-terminal helix of HACE1, shown here in yellow, and the N-lobe of the catalytic heck domain, shown in green. So zooming in on this interface, you can see here that the N-terminal helix is amphipathic. It has a side that's facing the subunit interface, which is composed of 3 adjacent leucine residues, leucine 8, 11, and 15, that appear to make hydrophobic contacts across the subunit interface. Now, so we expected that if we truncate this helix and make a shortened construct, which we call delta N, that this construct would be dimerization deficient. And indeed, we see that now if we determine the weight using mass photometry, we obtain a monomeric form of HACE1. We can also achieve the same effect when introducing point mutations at these sites. Here I show you data for replacements with aspartate, but we did similar experiments replacing the leucine with alanine. And the effect is also a shift from the dimeric state over to a monomeric form. The extent of the effect is obviously dependent on the position of the mutation, with the residues L15 and 11, which make more contacts in our structure, also have a larger effect on disrupting the dimer. The L8 mutation is more peripheral and consistent with this, the effect on dimerization is smaller. Does this translate to a cellular context? In order to study this, we performed co-immunoprecipitation experiments in cell lines that express either the full-length wild-type HACE1 or these dimerization deficient variants that we had characterized in vitro. And we're using here stable cell lines that are in principle inducible, but we chose not to induce the overexpression of the exogenous protein and to instead rely on a small amount of leaky expression in order to have lower levels of the exogenous protein, which you can see from the quantification here are about twofold the level of the endogenous HACE1. And so in this experiment, we are doing an IP of via the GFP tag of the exogenous HACE1, and we are monitoring the association with endogenous HACE1, which runs at a different height. And you can see here very clearly that the full-length protein has the capacity to self-associate, as shown by the band boxed here, whereas the dimerization deficient variants also show no self-association in this setup. So there is a nice correlation between our in vitro and these cell-based data. Now, we were curious also to study the conformational consequences of this dimer-monomer transition of HACE1. And to look at this, we made use of hydrogen-deuterium exchange mass spectrometry. This is a very valuable technique to us that we perform in collaboration with Dr. Wieland Steinchen at the University of Marburg, who is an expert in HDXMS. In brief, for this technique, the protein is subjected to a D2O solution, so containing deuterium, and given time for the amide protons to exchange against deuterons, then the exchange is quenched by a jump in pH, the protein digested, and the exchange pattern along the sequence read out by mass spectrometry. Now, we performed this type of experiment both on the full-length dimeric HACE1 and the delta N construct, the monomerized variant. And this allowed us to compare the two exchange patterns now and mapping the differences between these states onto the cryo-EM structure. What we're looking here at are changes that occur upon disruption of the dimer, and the changes that correspond to an increase in solvent exposure, so an increased exchange rate, are colored in shades of red. And this is what you can see here. We exclusively see an increase in HDX, an increase in flexibility and solvent exposure when disrupting the dimer. And intriguingly, these changes recapitulate the dimerization interface that our cryo-EM structure shows. So these are the largest changes are in dark red over here in the dimerization interface. And we also see propagated changes within the anchorin repeats, and intriguingly also within the HEC domain, specifically in hinges that we expected to be important for activity. HEC domains are known to require high flexibility for their catalytic cycle. And so the fact that the monomer appeared to increase the flexibility indicated that we might expect changes in activity when comparing the dimer and the monomer. And so we went on to characterize the activity of these states using first a reconstituted in vitro system where we mix the E1 enzyme, the E2 enzyme, fluorophore-labeled ubiquitin, the E3 HACE1, and a physiological substrate, RAG1, in the test tube, and we monitor the ubiquitination of the substrate RAG1 by way of the fluorescent tracer on ubiquitin, which we can monitor here nicely by SDS page. So what you see on the gel are reaction products, so ubiquitinated forms of RAG1. And what is immediately evident is that the full-length wild-type protein, the dimeric form of HACE1, is strongly auto-inhibited, whereas the dimerization-deficient mutants promote RAG ubiquitination very efficiently. The same effect can be observed in a cell-based setup where we transfected cells with the ligase HACE1 and GFP tagged RAG1 substrate. We pulled down RAG1 via the GFP tag and then plotted against ubiquitin. And once again, you can see here the dimerization deficient variants of HACE1 promote ubiquitination of RAG1, whereas the wild type is auto-inhibited. Now, to understand where this auto-inhibition comes from, we have to again look at our structure and specifically at the contacting site of the N-helix in the dimer. As I mentioned earlier, this site is made up by a portion of the catalytic N-lobe of the HEC domain. And specifically these hydrophobic residues marked here, which upon mutation to aspartate once again disrupt the dimer interface consistent with our structure. Now, this area, this hydrophobic area on the end lobe is a well-known region that is a conserved binding site in HECT-E3s for the upstream E2 enzyme. Domain derived from our cryo-EM structure and a modeled E2 based on a previous crystal structure determined by Brenda Schulman's lab of the E2 enzyme bound to a homologous HECT domain. And based on this conserved positioning of the E2 enzyme, it becomes very clear that this E2 clashes with the position of the critical N-terminal helix in the context of the HACE1 dimer. So dimerization should be incompatible with the productive interaction of HACE1 with the E2 and with the productive transfer of ubiquitin from the catalytic cysteine of the E2 over to the catalytic cysteine of HACE1. Now, we tested this using, again, an in vitro dedicated assay, this time a single turnover so-called discharge assay, where we monitor the loading of ubiquitin of the E2 enzyme onto HACE1. And again, this is a fluorescently labeled ubiquitin that we're following here via fluorescent gel. And one can appreciate here that the dimeric full-length HACE1 is unable to promote discharge of ubiquitin from the E2, whereas the delta N construct, the monomeric variant, promotes offloading of ubiquitin as expected. And now the mutations introduced at these hydrophobic residues of the N lobe that form part of the dimer interface, again, abrogate this activity as expected. And so what this boils down to is a model in which the dimeric form of HACE1 is auto-inhibited by preventing productive interactions with the E2. Activity of HACE1 requires release of the N-terminal helix and the disruption of the dimer, which then allows for the E2 to transfer ubiquitin over to the active site. A key question, obviously, is how this dimer-monomer transition of HACE1 is regulated. While we do not have a final answer for this, and we're actively, of course, working on the underlying mechanism, I would like to propose a hypothesis on a factor that may impact the dimer-monomer transition, and this is phosphorylation. So if we look at phosphoproteomic data on HACE1, we find that 3 phosphorylation sites coincide with the dimer interface we characterize. This is a tyrosine residue up here, threonine 20 and serine 14 around the critical N-terminal helix. As I explained, the dimerization interface is hydrophobic, so having phosphorylation at these sites would be expected to interfere with the formation of the dimer. To test this, we introduced phosphomimetic mutations, so glutamate substitutions at these 2 sites, serine 14 and threonine 20, along with a negative control, which is not reported as a phosphorylation site. And we had to use those mimetics as the kinase targeting these sites in cells is unknown. But as expected, when introducing a charge at these critical positions in the hydrophobic interface, we find that HACE1 becomes monomeric. Testing the effect of those phosphomimetic mutations in the same assay I introduced earlier shows, as expected, that the phosphomimetic variants are activated compared to the auto-inhibited wild-type protein. And the same effect can be seen again in a cell-based substrate ubiquitination setup where we monitor chain formation on REC1, which once again is enhanced for the mutated variants compared to the wild type. And so this suggests that a phosphorylation of HACE1 at the dimer interface may be a mechanism that would stabilize the monomeric state and prevent the formation of the auto-inhibited dimeric state. However, we also speculate that interaction partners and regulators may be involved, and they still need to be identified. Now, the work I showed you so far was done by a fantastic graduate student in the lab, Jonas Döring, who just graduated and is now a postdoc. He also created many of the figures on I showed you on the previous slides. If you take a look at our publication, you'll see we also worked out the mechanism of substrate recognition and specificity of HACE1, specifically its interaction with REC1 and the active GTP-loaded state of REC1. And we could explain how specificity arises from determining a cryo-EM structure of this complex that we're very excited about. This was a terrific work by Madita Volta, a postdoc in my laboratory, and I would highly recommend that you take a look at the publication to get a picture of the full story. And so with this, I would like to thank all the people who contributed to this work, Jonas, Madita, but also Camilo Torres and Julia Toblak, who were students in the lab contributing to this story. We're very grateful to fantastic collaborators who contributed to the work I presented today. In-house, I would like to acknowledge Christian Dienemann from the cryo-EM facility of our institute, who was really instrumental in helping us with the cryo-EM structures. Also Ulrich Steuerwald, Henning Urlaub, who is the head of the MESPAC group here in-house, and his postdoc Oleg Stupkov did the MESPAC analysis. As mentioned before, we're collaborating with Wieland Steinchen from the University of Marburg for the HDXMS analysis. Kate Bonsack was very helpful in the generation of stable cell lines. And I would also like to give special thanks to Clément Planchet from EMBL Hamburg, who helped us with SAX measurements and analysis, as well as Kim Remans from the EMBL Heidelberg, the head of the expression facility there. I'd like to thank you for your attention and happy to take any questions.